Welcome back to another video for IB Environmental Systems and Societies. Today we're talking about topic 3.1, an introduction to biodiversity. The first big idea in topic 3.1 is that biodiversity can be identified in a bunch of different forms. And those are species diversity, habitat diversity, and genetic diversity. You should remember all three of those terms. The idea here is that more habitats create more unique combinations of biotic and abiotic factors, which create more ecological niches, and more ecological niches provide opportunities for a greater diversity of species to live. Within a species, genetic diversity means that a range of individuals may be more or less suited for that unique combination of environmental factors within a particular niche or habitat. Biodiversity is a big concept that incorporates species habitat and genetic diversity. For the moment, I want to focus on two terms from topic 2.5 that you should already know, and if you don't, you should remember them now. The first is species richness. That's simply the number of different species found in an area. The second part of biodiversity is called species evenness, and that's how those species are distributed within an ecosystem. So here we have two different communities. They both have the same number of species. So they both have the same species richness. But this one on the left has a more even distribution of species. Not one of them dominates. Therefore, this habitat or this community on the left is generally considered to be more diverse because the community on the right is dominated by this one tree species that you see here. This image exemplifies the concept of genetic diversity. All of these carrots are the same species, but because they have different combinations of alleles in their genomes, they have different phenotypes or physical appearances. That means some of these carrots may be better suited for certain climatic or soil conditions. Most people think of biodiversity as simply the number of species in an area. And generally speaking, that's probably the broadest idea of the concept. Species diversity is both the number of different species, species richness, and their relative abundance or distribution across this ecosystem. That's the species evenness. One of the things that you learned about in topic 2.5 when we're investigating ecosystems is how to objectively measure biodiversity. That's where Simpson's diversity index or Simpson's reciprocal index comes into play. That is probably the best standard way to assess biodiversity in an area. Habitat diversity, as you might guess by the name, is the range of different habitats found within an ecosystem. On the screen, you see 4 different habitats in Brazil. The one on the top left is mostly open grasses. The top right has some grasslands and low-lying shrubs. Bottom left is a secondary forest. As you can see, it's quite bright. There's still a fair amount of sunlight coming through. The canopy isn't closed. And in the bottom right corner, you have the darker, more dense primary forest. Because these habitats have different combinations of abiotic factors, they support different species of both plants and animals. Again, we're going to come back to these two concepts of species richness and species evenness because they are important. You should be able to use both of these terms when discussing different components of biodiversity on your ESS exam. Remember, species richness is what most people think of when they think of biodiversity. It's the number of species in an area. Species evenness is how those species are distributed within an ecosystem. One of the reasons Simpson's index is so important is that we can describe communities of organisms using those diversity indices. Low diversity can tell us a lot about a particular area. It frequently indicates that there is pollution, such as eutrophication, or some other type of negative human impact on the biological components of an ecosystem. If we go back to the idea of succession in topic 2, we also know that diversity levels change throughout the course of succession. In different phases, there are different levels of biodiversity. So by being able to measure the biodiversity, we can assess where in the successional process an area is. I really like this image to exemplify the idea of habitat diversity. In this single watershed, where all of the precipitation that falls between these mountain ridges drains out through the watercourse in the middle, you have a really wide range of habitats. Right? We have bare rock here in the mountains. We have some low-lying plants in the mountains, coniferous forest lower down on the slopes. Here along the waterways, we have riparian zones. We also have these sand and gravel banks. And each one of those habitats has a unique combination of microclimate, physical conditions that make it possible for different plants and animals to grow there. So this is why habitat diversity leads to greater species diversity. Just like the carrot example earlier, genetic diversity results in greater variation within a single species. So all of these butterflies have different physical appearances as a result of the allele and genetic combinations in their genomes. Some of them are blue, some of them are red, some are some shades in between. The reason that genetic diversity and variation leads to greater degrees of biodiversity is because in different environments, different individuals have greater chances of successful reproduction. In this environment, the blue butterflies stand out against the background, while the red ones remain much better hidden. That means the blue butterflies are more likely to be eaten by predators, and the red butterflies are more likely to have successful reproduction and pass on their genes. This leads to speciation. Should the environment change, the reverse can be true. In this case, the blue butterflies are the ones who are well camouflaged and hidden, and the red butterflies are the ones that are most likely to be eaten. That means the blue butterflies are more likely to be able to pass on their genes and survive. Conservation efforts depend on our ability to effectively measure or assess biodiversity. If we go into the field and we can identify areas on our planet where there is a high degree of species diversity or genetic diversity within species or a high degree of habitat diversity, then we know where we should be concentrating our conservation efforts. This map shows biodiversity hotspots around the world. The different shades of red, orange, and yellow on the map indicate varying degrees of threats to biodiversity. The areas that are red are most impacted by human populations and see the greatest threat to the loss of biodiversity. If you notice in this infographic, you can see in this band of tropical rainforest all around the globe, this is where biodiversity is concentrated. That's because of the high levels of net primary productivity in tropical rainforest, those climatic conditions that allow plants to grow year-round. Singapore is found in what used to be tropical rainforest. You can see in this map from 1819, about 200 years ago, that Singapore was almost entirely primary rainforest and freshwater swamp forest. Then about 170 years later, the island of Singapore is almost exclusively urban and agricultural land with some patches of secondary forest in the middle. What that has meant for Singapore is that the vast majority of reptiles and amphibians are extinct or threatened with extinction. A similar pattern exists across Southeast Asia, again with amphibians and reptiles showing the greatest risk of extinction due to human impacts. You should remember that biodiversity really exists on three different levels. The broadest one is ecosystem or habitat diversity. The greater the range of habitats, the greater the range or variety of species that live in that area. Within a single species, there are, of course, differences among individuals, and those differences are the result of genetic differences. As we discuss the importance of assessing biodiversity, we need to remember that that assessment is only as solid as the quality of the data that we collect. Data describes patterns, and that data influences decision-making. Right? We can make better decisions if we have better data. So our ability to assess biodiversity depends on making reliable, repeatable investigations in the field. There are a number of factors that can affect the quality of the data that we can collect about biodiversity. Some places are harder to access and therefore aren't studied as well. Some species are harder to count because they're microscopic or they are challenging to find within the environment. The diagram you see on the screen shows many of the different threats to biodiversity in the Amazon rainforest. Not all of these threats are equal. Generally speaking, the bigger the circle, the greater the threat to diversity. And they don't always add up to 100% because many of those threats overlap. Generally speaking, when the forest is first opened up to logging, what then follows is agriculture. So these two factors work together as a threat to biodiversity. Having reliable data about the habitat diversity, species diversity, and genetic diversity within an area helps decision makers choose course of action, whether to proceed with a conservation project or a development project. The better the data, the more well-informed the process is, and hopefully humans will make the right decision. That's it for topic 3.1, Introduction to Biodiversity. Thank you for watching. Welcome back to a new video for IB Environmental Systems and Society. Today's video is about topic 3.2, Origins of Biodiversity. It's going to be a quick review to help you get ready for your mock exams and those big IB ESS examinations at the end of the school year. Let's get into it. First up is that evolution is a gradual change happening over many, many generations. It is a genetic change that leads to the changes in physical appearance or the phenotype of organism driven by a process called natural selection. This is Darwin's big idea, originally published in The Origin of Species in 1859. Evolution is probably the most heavily evidenced scientific theory in history. We have thousands of fossil examples from a wide range of species spanning several billion years, showing that it's been happening. We have genetic evidence in mitochondrial DNA. We can track mutation rates across generations. We can watch evolution happening in real time, both in laboratory experiments and with things like bacterial resistance to antibiotics happening in the world's hospitals. There is an overwhelming amount of evidence in support of evolution by natural selection. One of the first rules of the process of natural selection is that there is variation within populations, and you can see variation in the patterns of this giraffe fur here. These are all different species of giraffes. And the idea is that some of the variation benefits the survival of the organism, some of the variation harms the survival chances of the organism, and some of it has no impact. So the premise is that the variations that harm the survival of the organism tend to be filtered out because those organisms don't survive to reproduce. Whereas the characteristics that are genetically coded for that give that organism a survival advantage tend to be selected for, and their frequency in the gene pool increases over time. You don't need to know the details of natural selection like you would for an IB bio class, but you do need to know this general process. If you really want to get into the details of it, you can click on this link right here. I'll drop it into the comments of the video, and you can read all about evolution of the different shapes of bird beaks. The next big idea in this topic, it is actually the environment that drives the process of evolution. This isn't something that species choose to do. It is that the environment selects who lives and who dies. And the most classic example of this is the English peppered moth, which you can see here on the screen. In the English countryside, before the Industrial Revolution, the peppered moth liked to live in our birch trees, which have this pale gray bark. Pale gray peppered moths found it easy to be camouflaged there, and pepper moths with the darker pigmentation were more readily seen and eaten by predators. Then we started burning fossil fuels and leaving soot on everything in the countryside, which covered our nice pale birch trees in a dark layer, which then meant that the lighter colored moths began to stand out against the background, and the darker colored moths were more easily camouflaged. So the predators switched from eating the dark moths to eating the light moths. Again, the moths didn't choose to evolve, but the environment changed and applied a different or new environmental pressure on the moths. So there was a shift in the gene pool. This happens over many millions of years, happens over many generations, and what's going on is if the environmental pressure is great enough on one population, it will eventually become a new species because the genes will have shifted enough that that population can no longer successfully interbreed and produce fertile offspring. The most famous example of this, and what really prompted Charles Darwin's thinking, is the Galapagos finches. When Darwin traveled there in the mid-1800s aboard the HMS Beagle, he noticed that there were species of finches on all of the islands, and that the beak shape of the birds was correlated with the type of food
on each island, because each island has a different set of abiotic factors and therefore different environments. The Galapagos Islands are really isolated way out in the middle of the ocean, and so this idea of adaptive radiation is that there was one original species of finch that came to the islands, and as its offspring survived, the ones who had beak shapes through their variation that were particularly adapted to finding food on certain islands gradually developed physical and behavioral differences until they became new species. So the new species adapted to new environments and radiated out from the original species. Generally speaking, speciation is driven by isolation. You will have a population of organisms that can go back and forth between two bodies of water, for example, when sea levels were higher. And then as the sea levels dropped and all of this land in Central America suddenly became above sea level, these populations could no longer interact and interbreed. Because that changed the movement of water in the oceans, it changed the ocean currents, the environment changed in the Caribbean compared to the Pacific, and over many generations, the porkfish split off from the Panamic porkfish. These used to be one species, but after the appearance of the Central American peninsula here, these two populations were no longer able to interbreed, and they eventually became totally separate species. Although they are related, you'll notice they still have the same genus. You should be able to explain how plate activity has influenced evolution and biodiversity on Earth. The big ideas here are that land masses continually move around our planet over many millions and billions of years. And as they move, they might migrate from the tropical zones into the temperate areas or into polar zones. And that as they move around, migration routes change, because land masses that were connected suddenly become disconnected. Land masses that were separated are then joined together. And so that changes the way that different populations of organisms interact with one another. You don't need to know the details of Earth's inner structure and be able to explain the details of tectonic forces driving the movement of Earth's plates for ESS. What you do need to know is that as those land masses move in and out of different climate zones, and as Earth's climate changes through ice ages and warm spells, that makes migration routes available for species to interact, and it breaks migration routes, further isolating species, which then, of course, influences the genetic makeup of subsequent generations. Some of the evidence that we have for evolution is that we have fossil remains on land masses that are currently very far apart from the exact same time period, showing that in the past those land masses were together. Here we have the Cynognathus reptile. Its fossils are found in South America and Africa. We have these Glossopteris ferns whose fossils are found across all of the continents in this image. And if you can recall, Antarctica right now isn't really a place where you're going to find too many ferns, primarily covered with a big thick ice cap. The fact that we have fossils of the same species at the same time across all of these land masses shows that in the past all of these land masses were at one point connected, and these species could migrate back and forth. One of the big ideas you need for ESS is to know that there have been in Earth's past what we call mass extinction events, and they're indicated here by these yellow triangles. And you can see where the number of species on the planet drops very precipitously every time we have one of those mass extinction events. Why do those mass extinction events happen? Why does biodiversity fall off periodically in Earth's history? Here are the major mass extinctions that have been documented so far. Names or the timings of them. I suggest you probably remember the KT event that happened 65 million years ago, knocking out the dinosaurs. But if you look at these causes, rapid global cooling, rapid global cooling, rapid global warming, all of these mass extinction events have been driven by rapid climate change. And that's one of the reasons why climate change is such a big deal today. As a matter of fact, extinction rates today have spiked in a way that they have during all 5 of Earth's past mass extinction events, leading many scientists to conclude that now, in the time of people, called the Anthropocene, we have actually entered the 6th great mass extinction event in Earth's history. That's it for our review of topic 3.2. I hope it helps you prepare for your mock exams and your final exams. I wish you the best of luck on your exam and happy learning.
Welcome to another video for IB Environmental Systems and Societies. Today's topic is 3.3, Threats to Biodiversity.
I'm going to run through this relatively quickly to help you review for your mock exams and prepare for the IB ESS examinations coming up at the end of the year. Let's get into it. The first big idea is that global biodiversity is difficult to measure exactly, but we are aware that it is decreasing rapidly due to human activities. One of the tools that we use is assigning species conservation status, and that's done through the IUCN, the International Union for the Conservation of Nature. Quantifying biodiversity. Estimates of how many species there are on our planet vary widely. And if we go down here and we take a look at what's hiding back here behind my picture, we see that 86% of all species on land and 91% of those in the seas are yet to be discovered. In part, that's because they're difficult to find, in part because of a lack of funding. There are many reasons for that. But basically, the short answer is we're not entirely sure what lives on our planet. We've only assessed a small portion of it. We also know that extinction rates are increasing rapidly. In my previous video about origins of biodiversity, we looked at the background extinction rate, and we know that current rates of extinction far exceed the historic background extinction rate. Most of those extinctions are due to habitat destruction as the human population expands into previously undeveloped areas, the introduction of invasive species as our global commerce becomes increasingly interconnected, the addition of pollution to the ecosystem, and overharvesting and hunting, both of which are kind of the same thing. Current rates of extinction loss, really, really high compared to where they've been. We're looking at possibly the world's sixth great mass extinction right now, called the Anthropocene. If we look at the red list, that's the IUCN red list showing extinction rates, right? The groups of species that are most at risk of extinction here. Oh, wait. Better odds. Look at the corals. Really clearly stands out here. Corals are dropping very rapidly because they exist only in a very narrow band of optimal temperature and aquatic conditions in shallow seas. As the ocean rises due to climate change, and as those areas are increasingly subject to human interference activities, those corals are increasingly stressed. Then we get this massive statement from the ESS syllabus: red list, population size, speciation, distribution. These are all the things that are factors we use to determine the conservation status of species. So instead of trying to do this all at once, let's look at them one by one. Okay. Here on the left side of the screen, you see the IUCN labels. Least concern, as you might guess by the name, we're not terribly worried about the status of these species. They seem to be doing just fine in face of human activities. The farther down this list you go, the closer species become to extinct until they actually become extinct. So extinct in the wild means the only place that they exist on our planet are in captivity. Critically endangered, and vulnerable are all considered threatened. All right? You can see those here, red, orange, and yellow. So as we go down the list on the left or up the list on the right, the risk of extinction increases. What are the factors that go into that? Well, they're pretty varied. But if we look at this list here, and let me move myself out of the way for a moment. If we look at this list here, 2018, right, of all of the gastropods, almost 2,700 species. Almost 2,700 species of gastropods. Most of them are green or least concern, similarly with the bony fishes, right? So lots of green is good. The more red, orange, and yellow you see, the more threatened those species are as a portion. So if we go to the bottom of the list here, the cycads, which are an ancient palm-like tree that's related to ferns, of the 307 known species on Earth, more than half of them, about 62% of them, are threatened in some way. The IUCN uses several different factors to consider whether or not a species is threatened or how threatened it is. Population size. It's easier to wipe out small populations. Species that are highly specialized for a very particular ecological niche also at risk of extinction, because if that niche gets modified or wiped out, so does that species. Species that are distributed very widely, because they're in a wide range of habitats or ecosystems, are less likely to go extinct because the damage would have to be extraordinarily widespread to remove them from our planet. Reproductive potential. That refers to reproduction rates. So the more offspring a species has, the lower their risk of extinction. That's why you're going to see things like cockroaches and ants and bacteria at the bottom of your extinction lists in terms of risk levels. When we look at geographic range and how that range is fragmented, right? Intact habitats allow organisms to migrate in and out and allow populations to intermingle and interbreed. We talked about that a little bit in topic 3.2 in biodiversity, right? So the more intact the habitat is, the lower the range, the lower the risk of extinction. Again, also habitat that is high quality and not degraded, that provides ample nutrients and energy pathways to counteract disturbances by people, right? And trophic level, right? The lower the trophic level, the lower the risk of extinction. The higher the trophic level, frequently the higher the risk of extinction. And last but not least, we combine all of those factors to reach a determination of conservation status. Right? Tropical biomes contain some of the most biodiverse areas because of their high primary productivity due to Earth's climate. They're also very popular areas for people to live, and because of that, they are being unsustainably used or exploited, and because of that, we have massive losses in biodiversity. Tropical biomes are some of the most diverse biomes on the planet. Tropical rainforests are considered the most biodiverse biome on Earth. And they are, unfortunately, because of their wealth of species, also a wealth of resources. And so they are being exploited and have been exploited for many years. That means in this band of tropical rainforest that you see in green across this map, that we have potential for lots of conflict. And here you are in DRC, one of the wealthiest countries on Earth in terms of resources, and yet one of the poorest countries on Earth in terms of economic development. One of the things you need to be able to do in ESS is discuss the case histories of 3 species. And I've got a few case studies on my website if you want to check out. You need one that is already extinct due to human activities. You need a second one that is currently critically endangered. And you need a third one that has previously been threatened, but whose status has improved due to conservation efforts. That doesn't mean they're no longer threatened. It just means that they've become less threatened. I encourage you to check out some of the case studies on my website because students put those together and they're pretty good. Then, of course, you need to describe threats to biodiversity from human activities in a given natural area. So you might want to discuss the Congo rainforest basin or the Amazon rainforest, or you could discuss the Great Barrier Reef in Australia. The named area, the named natural area, does not matter as long as what you can do is describe what the threats are to biodiversity in that area. Then you'll also need to be able to evaluate the impact of human activity in tropical biomes. all of these little dots here show different threats to human activities. So these two slides, these two activities, describing threats to biodiversity in a given natural area and evaluating the impact of human activities, those are very closely connected. So look at what people are doing in tropical biomes, pick an activity, and describe that threat in that area. And that will probably lead you into a discussion of conflict between exploitation, meeting the needs of people, sustainable development, while ensuring that those needs of future generations can still be met, and conservation, right? That there are conflicting priorities for all three of those goals. That's it for our quick review of threats to biodiversity. I hope you found this review helpful, and I hope to see you next time. Happy learning.
Welcome to another IB Environmental Systems and Societies video. Today's topic is 3.4, Conservation of Biodiversity. This will be a quick review to help you prepare for mock exams and those official examinations at the end of the school year. Good luck. Let's get into it. The first significant idea here is that the loss of biodiversity is what's driving efforts to conserve it, and those efforts to conserve biodiversity are greatly influenced by one's environmental value system. The variety of arguments given for the conservation of biodiversity, of course, depends on those environmental value systems that we've discussed back in topic 1. If you look at this graphic, how much tropical forests absorb and store carbon, is this more of an anthropocentric or a technocentric or an ecocentric approach? You should be able to justify whatever conclusion you come to based on evidence or scientific reasoning. The next big idea in ESS topic 3.4 is that there are differing approaches to the conservation of biodiversity. You can see those here on the slide. You're either going to target the habitat or focus on specific species. Arguments about species and habitat preservation can be based on aesthetic, ecological, economic, ethical, and social justifications. Again, the ecological perspective argues that rare or endemic species may be lost and that it is necessary to conserve the habitat or species because of their role in food systems and productivity. From a technocentric perspective, maybe argue from the economic point of view about ecotourism and medicinal compounds that are derived from plants. Those are some of your ecological goods that we derived from protecting habitats. There's also the ethical argument about biorights that skews towards the ecocentric end of the spectrum. And then, of course, the social argument for conserving biodiversity is that habitats provide homes, work, resources, and social cohesion for indigenous people. That will be somewhere in the ecocentric to anthropocentric end of the EVS spectrum. You should be able to discuss how different governments or organizations use different tools or strategies to promote the conservation of biodiversity. The logo here on the screen, this panda, is probably the most famous logo in the world after the McDonald's Golden Arches. This shows that WWF has mastered the use of media to motivate people for biodiversity conservation. There's a range of government and non-government organizations involved in conservation of biodiversity. The International Union of Conservation of Nature, IUCN, publishes the Red List, which is considered the comprehensive guide to the conservation status of many species on Earth. And that list is used to guide decision making. Different organizations have different degrees of effectiveness. Big, gigantic organizations such as the United Nations have a global spread and influence, but they're also quite bureaucratic, which restricts their effectiveness. Smaller grassroots organizations have a much smaller scale of operation, but because they are small and they operate only in one or two areas, they tend to be locally effective, but minimally effective on a larger scale. International conventions on biodiversity create collaboration among nations. One of the most famous conventions that you'll encounter in the ESS syllabus is CITES, the Convention on the International Trade of Endangered Species. This came into effect in the 1970s and has actually been quite successful, particularly in combating the poaching of elephants and rhinos in East Africa. One of the things you'll probably be asked to do on an ESS exam, either in paper 1 or paper 2 questions, is to discuss or evaluate habitat-based approaches versus species-based approaches to biodiversity conservation. The habitat approach focuses, as you might guess, on protecting the entire habitat and all of the species that live there. The species-based approach is very narrowly focused on target species, generally ones that are very critically endangered or near extinction.
Both approaches have benefits, both approaches have limitations, and so most conservation efforts fall somewhere in the middle where we call a mixed approach, using the strengths of each to offset the weaknesses of the other. Perhaps in a case study for paper 1, you might be asked to evaluate the strengths and weaknesses of a named protected area. And there are many factors we use to evaluate protected areas. One is the size of the area. A large area is better than a small area. The shape of the area, as you can see on the left side, this is a more round shape. That reduces what we call the edge effect. The edge effect is where the core protection zone is close to or interacts with areas of human habitation or areas with greater degrees of human influence. A big area is better than a small area. A round area is better than a long, narrow strip. An area that has not been split or fragmented by roads or infrastructure are better. The fewer disruptions to the core, the better the efforts at conservation. Criteria for evaluating protected areas include looking at the shape of the protected area. The closer the area is to being round, the better it is, because the perimeter or the edge effect is minimal with a round shape. When you have long, narrow, skinny shapes, the core becomes very small and the edge space becomes quite large. If it's not possible to design one big core area, the next best approach is to design several smaller core areas and connect them through corridors to allow wildlife to migrate back and forth between the protected areas. Species-based conservation strategies. The CITES Convention falls into this category because it identifies species by name. Species that fall within the vulnerable, threatened, endangered, critically endangered categories of the IUCN Red List show up on the CITES list. They're categorized into Appendix 1, 2, or 3 based on the degree of threat. And that convention prohibits international trade in those very specific cases. Here you have the California condor that is part of a captive breeding program that's actually been quite successful. California condors were almost entirely wiped out in the 1980s. So conservation biologists took a bunch of eggs from their nests, they brought them into secure areas, and they raised condors from hatchlings to maximize the care and protection that they could get. You'll notice also there's a hand puppet from the human on the left side that looks like a mama vulture feeding the condor. You'll also notice the hand puppet on the left that looks like a mama condor feeding the chick condor. That's so the chicks don't become acclimatized or used to human presence. This helps boost the success of their reintroduction in the wild, which is one of the frequent criticisms of species-based conservation, because often animals that have been reared in close contact with humans lose the ability to hunt on their own and fend for themselves in wild situations. So the success of their reintroduction into wild habitats tends to be quite low. Reintroduction. The idea here is that we remove individuals of the target species, we bring them into a protected area, raise them, increase their populations, and then take them back to their original habitat and release them into the wild. It sounds good in premise. It tends to be quite expensive, and it tends to have mixed results because it doesn't directly address the habitat loss, which is frequently one of the primary factors that the species is threatened in the first place. Species-based conservation is frequently based on the use of a flagship species. This is like the WWF logo with the panda on it. A flagship species is one that is considered charismatic or famous. It draws people attention. It makes people feel warm and fuzzy, but that's different from a keystone species. A flagship species is famous, but a keystone species is critically important for the functioning of an entire ecosystem. A flagship species might be a keystone species, but generally speaking, if a flagship species disappears, the entire ecosystem isn't going to collapse. Whereas if a keystone species disappears, the ecosystem is at risk of collapse because the food webs will fall apart. When asked to evaluate conservation efforts, one of the things you have to look at is how they benefit people, because let's face it, if people don't benefit from conservation efforts, they will fail. People will look out for themselves first. We've seen that pattern throughout human history. So if the surrounding communities don't benefit from the conservation effort, a protected area is likely to enjoy less success. The location of a conservation area is also an important factor when evaluating the success of that area. This is an image of Nairobi National Park right outside of the biggest city in Kenya. The benefits of it being that close are that people have regular contact with the animals and they become aware of the conservation status of those animals. The drawback is that people have regular contact with the animals, and that threatens the animals because sometimes there are accidents on the road with vehicles. Those animals may stray beyond the boundaries of the park, in which case they're encroaching on human settlements, and that leads to human-animal conflict. You may be asked to explain the criteria used to design and manage protected areas. Here we have a bunch of protected areas in Nepal, the blue ones along the northern and northwestern borders. You see one in the middle near Annapurna, is quite large. Simply from its size and its almost round shape, you might conclude that that could be a very successful protected area. Then if you look in the southwest near Bardia and Banke, you have two long, skinny conservation areas, which means they have a lot of edge effect, a small core zone, and if you look at where they are on the map, they tend to be far away from other conservation areas, which inhibits the migration of animals in and out of those zones. As part of your ESS case study in paper 1, you might be asked to evaluate the success of a named protected area, or it could be part of one of those structured essays in a paper 2 question, or it could be part of one of those paper 2 structured essay questions that are used those 9-mark bands. If you're asked to do that on paper 2, you need to be able to consider the size, the shape, the edge effect, corridors, the proximity to human settlements, the fragmentation of the habitat, and the benefit of the local community when determining whether that area is or is not, or may not be successful. Pretty much every year, you're going to encounter some kind of a question about evaluating habitat-based approaches to conservation versus species-based approach to conservation. Broadly speaking, habitat-based approaches will be better because they're more holistic and they actually protect the entire ecosystem and more than just one species, so they conserve all biodiversity or a larger proportion of biodiversity, whereas species-based conservation may benefit one or two target species, but tend to be more expensive and overall less successful in the long run, unless also combined with habitat-based conservation efforts. That's it for the quick review of ESS Topic 3.4, Conservation of Biodiversity. I hope you found this video helpful as you prepare for your exams. Good luck and happy learning.